Have you ever wondered how scientists tell different bacteria apart? It's a fascinating process that goes beyond just looking at them under a microscope. They're far too small to see with the naked eye, let alone distinguish. Even under magnification, they can look quite similar. That's where the ingenious Gram stain comes in. This technique allows scientists to categorize bacteria into two main groups. Developed by Hans Christian Gram in 1884, this technique is a cornerstone of microbiology. It revolutionized the way we study bacteria. The Gram stain is a differential staining technique, meaning it uses dyes to differentiate bacteria based on their structural differences. This method is both simple and effective. It's like a magic trick, but with science. The process begins with staining the bacteria with crystal violet dye. Bacteria are first stained with crystal violet, then treated with iodine. This step is crucial for forming a complex within the bacterial cell wall. This forms a complex that gets trapped within the bacterial cell wall, making it easier to identify them later. Next comes the crucial step, a quick wash with alcohol or acetone. This step differentiates the bacteria based on their cell wall properties. Some bacteria hold onto the crystal violet stain tenaciously, appearing a vibrant purple under the microscope. These are known as gram-positive bacteria. These are the gram-positive bacteria. Their thick cell walls retain the violet stain. Others, however, lose the initial stain and are counterstained with a pink dye called safranin. These are the gram-negative bacteria. These pink-hued microbes are the gram-negative bacteria. Their thinner cell walls do not retain the violet stain. This simple color difference reveals fundamental variations in their cell wall structure, having profound implications for their behavior and our ability to combat them. Understanding these differences is crucial for developing effective treatments. The striking difference in how gram-positive and gram-negative bacteria react to the Gram stain lies in the architecture of their cell walls. Imagine the cell wall as a protective fortress surrounding the bacterium. Gram-positive bacteria boast a thick, sturdy wall primarily composed of Peptidoglycan. Picture a tightly woven, multi-layered mesh. This robust layer readily retains the crystal violet iodine complex, hence the purple color. Gram-negative bacteria, in contrast, are architectural rebels. They have a thinner Peptidoglycan layer, but with a twist, an additional outer membrane encasing the Peptidoglycan. This outer membrane, however, is not a fortress wall. It's more like a selectively permeable fence, controlling what enters and exits the bacterial cell. This structural difference, a seemingly minor detail, has major consequences. It influences everything from antibiotic susceptibility to the way these bacteria interact with our immune system. The outer membrane of gram-negative bacteria acts like a formidable shield, often making them more resistant to antibiotics compared to their gram-positive counterparts. This outer membrane is a complex structure composed of lipopolysaccharides, which provide an additional barrier to many harmful substances, including antibiotics. This is a growing concern in the medical community, as antibiotic resistance poses a significant threat to global health. The rise of antibiotic-resistant infections means that common treatments are becoming less effective, leading to longer illnesses and higher mortality rates. Think of antibiotics as keys trying to unlock and disable bacterial cells. However, when these keys cannot find the right lock, they fail to work, leaving the bacteria unharmed and free to multiply. The thick Peptidoglycan layer of gram-positive bacteria provides some resistance, but many antibiotics can still penetrate and do their job. This is because the Peptidoglycan layer, while robust, does not have the same complex outer membrane as gram-negative bacteria. The outer membrane of gram-negative bacteria, however, is a master key blocker. It effectively prevents many antibiotics from reaching their target sites within the bacterial cell. It possesses specialized proteins called porins that act as gatekeepers, controlling the passage of molecules. These porins can be highly selective, allowing only certain molecules to pass through while blocking others. Many antibiotics are simply too large to squeeze through these porins, rendering them ineffective. This size exclusion is a critical factor in the resistance mechanism of gram-negative bacteria. Furthermore, the outer membrane can house enzymes that degrade or modify antibiotics, further reducing their effectiveness. These enzymes can break down the antibiotic molecules before they can reach their targets. This inherent resistance makes treating infections caused by gram-negative bacteria particularly challenging. As a result, new strategies and treatments are urgently needed to combat these resilient pathogens and protect public health. Section 4. Friends and foes, gram-positive and gram-negative examples. Both gram-positive and gram-negative bacteria encompass a vast and diverse group of microorganisms, some beneficial, others pathogenic. Among gram-positive bacteria, we encounter familiar names like the culprit behind staph infections and a common cause of pneumonia. But don't let their disease-causing potential fool you. Gram-positive bacteria also include friendly faces like the species, crucial for fermenting yogurt and kimchi, and the genus, important members of our gut microbiome. The gram-negative camp boasts equally diverse members. A common resident of our intestines can cause food poisoning in certain strains, and notorious for causing foodborne illnesses also fly the gram-negative flag. And let's not forget the bacterium responsible for the devastating diarrheal disease cholera. Section 5. The clinical battlefield, gram staining in action. In the clinical setting, the gram stain is an invaluable tool for rapid diagnosis and treatment decisions. It allows medical professionals to quickly identify the type of bacteria causing an infection, which is crucial in emergency situations. Imagine a patient rushed to the hospital with a severe infection. The clock is ticking and every second counts. The medical team must act swiftly to determine the cause of the infection. Doctors need to act quickly to administer the appropriate antibiotics. The gram stain provides a rapid and reliable method to guide their decisions. A sample of blood, urine, or tissue is collected and subjected to the gram stain. This simple yet effective technique can be performed in a matter of minutes. Within minutes, the color revealed under the microscope provides crucial information. The results can indicate whether the bacteria are gram-positive or gram-negative. If the bacteria are gram-positive, certain antibiotics, such as penicillin or methicillin, might be the first line of defense. These antibiotics are specifically effective against gram-positive bacteria. If, however, the culprits are gram-negative, a different arsenal of antibiotics like ciprofloxacin or gentamicin may be necessary. These antibiotics are tailored to combat gram-negative bacteria. Knowing the gram status of the infecting bacteria allows doctors to target treatment effectively, improving patient outcomes. This targeted approach minimizes the risk of antibiotic resistance. The gram stain is a testament to the power of simple yet elegant scientific techniques. It has stood the test of time and remains a cornerstone in microbiology. It provides a window into the microscopic world, allowing us to distinguish between different types of bacteria and understand their unique characteristics. This differentiation is vital for effective treatment. This knowledge is crucial for combating bacterial infections, developing new antibiotics, and appreciating the intricate interplay between these microscopic marvels and our own health. The gram stain continues to be an essential tool in the fight against infectious diseases.